Okay, so let's look at some past paper questions. A common question that's going to come up is when they give you a diagrammatic representation of the DNA double chain. And it asks us to label the various components. And here we've got a base. Here we've got our sugar. It's actually a pentose sugar, or the actual name of it is deoxyribose. And then here we've got our phosphate. Easy. Okay, and here we've got the question, name the bonds that join the bases. A good guess in biology, if you didn't know, is hydrogen bonds. They pop up over and over again, and that is actually the correct answer. Okay, next question. Ribonuclease is an enzyme. It is 127 amino acids long. Wow, that's a long protein. What is the minimum number of bases needed to code for this enzyme? Well, let's just have a little look at the question for a minute. Ribonuclease, we know it's an enzyme because it ends in A's. And although we've never heard of ribonuclease before, it doesn't matter. It's an enzyme, so it will have the same basic principles. In actual fact, it's just asking us, basically that question may as well ask, how many bases code for an amino acid? And we know there are 3 bases that code for one amino acid. So I've got 127 amino acids, I need to times that by 3. And our answer is 381. Okay, just have a read of this question and then start and see if you can answer it. Okay, let's work through it. We know that 3 bases code for one amino acid. So let's split our whole code up into the 3 bases. So there's 1, there's 2, there's 3, 4, 5, 6, and 7. Now I know that amino acid Y, there's only 1 of them, and that's already been put into the table for me. And I can count my other amino acids. So this one, there is only 1 with that base sequence. This one, TAC, that appears twice. This one appears 3 times. And this one just appears once. So, therefore, I've got my Y in there. Then I've got the ones that come twice, TAC and TAC. So that must be X and X. Then I've got the one that comes 3 times, one after the other. So that must be this one, W, W, and W. And then the last one on the last base sequence is Z. So that will go in there. Okay, this question can pop up a lot. Explain how a change in the DNA base sequence could result in a non-functional enzyme. Well, remember we need 3 base sequences to code for one amino acid. This amino acid, for example, could be coded for by GAC. This amino acid could be coded for by TAC. This amino acid could be coded for by TTA. Now I'm making that up, but the point is still the same. How the hydrogen molecules stick out from that dictates how it's going to twist and fold. And that folding is really, really important because that's what holds it in its shape. Its shape is important because, of course, an enzyme has an active site. If the shape is different, the active site will be different, which means it won't be complementary to the substrate anymore. They won't fit together. It will be non-functional. For 3 marks, pause the video and see if you can structure an answer to that. So how did you do? Well, the first thing we need to say is that any change in base sequence would change the sequence of our amino acids. It would change the sequence of our amino acids because it might create a new one, put a new one in there. So this would affect the folding of the protein. If we fold the protein differently, then the active site will be a different shape. And that means that the substrate won't be complementary to it anymore. And that means our enzyme will be non-functional. So our marks would be change the sequence of amino acids, 1 mark. The folding of the protein would be different. At A level, we call that the tertiary structure. So the primary structure is the order or the sequence, and when it's all folded back on itself, that's the tertiary structure. And therefore, the active site may not be complementary to the substrate. That would be our 3rd mark. Here's another question that pops up again and again. I've used easy numbers here so we don't have to tax our brain too much, but basically the question asks, if adenine is 30% of your bases in a length of DNA, what would the others be? Well, if adenine is 30%, then thymine must also be 30% because they connect together. So for every one adenine, you've got a thymine. So if that's 60% of our DNA, there's 40% left. That 40% is made up from cytosine and guanine. So therefore, it must be 20% and 20%. You might not receive easy numbers like that, but it's basically the same principle. Last question, I promise. But these questions are particularly important because before they always appeared on the A level and now they're in the GCSE. And so there's no legacy material, there's no material left over from previous exams that contains questions like this. It says, name the 2 substances that make up part X on the DNA molecule. Okay, so in this case, we know that part X has actually got a lot more detail in it than it's shown there. In the diagrammatic representation we've got here, it's a straight line. But we know that the backbone to DNA is a sugar phosphate backbone. And so what we're going to say is we can say sugar and phosphate. We can be cleverer and we can say pentose sugar, or we can name the sugar and say deoxyribose. And phosphate is just phosphate. These questions are really important because this is new material in the GCSE. It used to be in the A level and now it's moved down. So we don't have the material, we don't have the past paper questions. All I'm doing is using A level ones in your GCSE. If you are interested in those higher grades, the 7, the 8 and 9, for single award biology, you really need to be able to manipulate the information. When I say manipulate the information, I don't just mean know it. We don't want just recall. We don't just want the names of things, which is why AQA are happy for you to remember them as A, C, T and G. What they want to know is, can you work with that information? So like in the penultimate question that we just had, if you've got 30% C, you would know that you've got 30% G and 20% A and 20% T. You're working with the information, and that's what AQA want. So please make sure you take some notes from this video and please review it again before you actually take your exam.